The overall goal of this procedure is to determine the presence of multiple cyanobacterial strains in liquid and solid samples using a fluorescence microarray immunoassay.
This method can help answer key questions in the environmental monitoring and water management fields, such as the presence of toxin-producing cyanobacteria before the blooms become visible to the naked eyes. The main advantage of this technique is that multiple cyanobacterial strains and multiple samples can be monitored simultaneously in a 3-4 hour period, both in situ or in the laboratory.
So this method can provide insight into biodiversity. It can also be applied to other fields such as monitoring aquaculture installations, life detection in extreme environments, or planetary exploration.
Generally, individuals new to this method will not struggle because of the simplicity in the sample processing and the ease of the interpretation of the results.
After producing and purifying antibodies according to the text protocol, add 100 microliters of DMSO into a commercial vial containing dye for labeling 1 milligram of protein. Once the dye is dissolved, label the purified antibodies by adding 2 microliters of the dye to 50 microliters of each antibody preparation. Maintain the labeling reactions at ambient temperature under continuous agitation on a vibrating platform for 1 hour. After purifying the labeled antibodies according to the text protocol, load a small aliquot into a spectrophotometer and measure the absorbance at 280 and 650 nanometers. Calculate the labeling